Hi everyone, we're going to focus on muscular system next. This is a new chapter for us and pretty much the first time we're going to encounter heavy-duty physiological topics. Let me start you off in our course pack on page 76. And we will first talk about the four major functions of skeletal muscle. We know that skeletal muscle provides movement in conjunction with moving our bones. Muscles also help us to maintain posture against gravity. They help us to stabilize our joints. And very importantly here, they help us maintain a body temperature by generating heat. We know that mitochondria make ATP from glucose and oxygen, and when that ATP is used, we can actually generate heat. So there is an involvement here in thermoregulation. Let's talk about the characteristics of muscle tissue. We will use these terms as we get further into this chapter. First is excitability. That is the muscle's ability to react to a nerve stimulus. If a muscle is not excitable, that means there is some difficulty getting the stimulus to reach that muscle from a motor neuron. So excitability is a good thing. It means that muscle can respond to a nerve stimulus. Contractility is a potential of shortening that muscle. In this picture, you can actually see a muscle shorten upon stimulation. So contractility is key for meaning that the muscle not only can receive the stimulus, which is excitability, but also contract and shorten. Muscles should also be elastic, meaning that they can return to their normal shape after shortening and stretching. So this is a key feature for how muscle tissue, and especially the heart tissue, must work. In your course pack, there are three pictures. On the far left is cardiac muscle tissue. In the middle is smooth muscle tissue, and on the far right is skeletal muscle tissue. You may recall that cardiac muscle tissue and smooth muscle tissue are both involuntary muscle tissues, while skeletal muscle tissue is a voluntary muscle tissue. Skeletal muscle is also striated, so striations are shared by skeletal and cardiac muscle tissue. Smooth muscle tissue, however, is unstriated or not striated. You may recall having looked at cardiac muscle tissue previously in our histology studies. You may recall that these are branching muscle cells, and they also have these intercalated discs, which are gap junctions that allow nerve impulses to travel directly from one cell to the next. So cardiac muscle tissue uniquely has these intercalated discs and the branching pattern. Where do you find cardiac muscle tissue? In the heart. All right, so just to look at this branching pattern that you can see here and some of these nice intercalated discs. Smooth muscle tissue lacks striations. This one, of course, is found in the walls of hollow organs like our digestive tract, urinary system tract, and reproductive tracts. Don't confuse this one with dense regular connective tissue. It does not have those wavy collagen fibers as you might see in, say, dense regular connective tissue. Now, this tissue works by a type of rhythmic contractions that we call peristalsis, and that is an involuntary function of helping to move items through these tubes, whether it's food through the digestive tract, urine through the urinary system, or even reproductive cells or a baby out of the reproductive tract. So you can see actually two layers of smooth muscle here. There's a circular layer and a longitudinal layer in a lot of these organs. We'll talk more about this as we get into Bio 211 and talk about how these hollow organs are set up with these rings and longitudinal layers of smooth muscle. Finally, skeletal muscle is striated as evidenced by the dark and light banding patterns here, and skeletal muscle also has a multinucleate setup. When skeletal muscles form, they actually fuse together multiple cells. Those multiple cells form what's called a skeletal muscle fiber. So when you bring multiple cells together, you bring along their nuclei. And in fact, these are multinucleate cells. So there's several sitting right there in that one cell. Think about this. The rectus femoris muscle that runs over your thigh is the length of your thigh, and that muscle contains cells or fibers that run the length of your thigh. So that could be, say many inches long, 18 or so inches that form the length of your thigh. So here's another look at skeletal muscle fibers. They are in fact fused muscle cells, and they do have this multinucleate characteristic. We don't tend to see that in other types of muscle cells. Great view here of these nuclei. You can see them here forming this skeletal muscle cell. So let's now turn over to page 77 and talk about the anatomy of a skeletal muscle. At the very top of your page, we're going to talk about the macroscopic anatomy, in other words, what you can see with the naked eye. Then we will work our way down to the microscopic anatomy at the bottom of your page. There are layers of connective tissue that form membranes around our skeletal muscles, and the outermost membrane is called the Epimysium. That is the blank for number 1. It is this term here, the Epimysium, which forms the outermost covering around the outside surface of your muscle. Let's take a look inside your muscle. Inside your muscle, we see these bundles of muscle cells, like this one here or this one here. Each one of those is encircled or encapsulated by number 2, Perimysium, this term here. So epi is the outer covering, peri wraps around fascicles, which are bundles of muscle cells. And finally, number 3 is Endomysium, this term here. And this, of course, means inside muscle covering. This particular kind of connective tissue covers individual muscle cells. So let's label this picture. On the outer surface, we have the Epimysium. That's your blank for number 1. Epi meaning above or upon. That is the outer covering here. We then have bundles. This is a bundle here. The bundle is called a fascicle, and the fascicle is actually surrounded by our number 2 blank, and that is Perimysium. Here we have an individual muscle cell pulled out. The muscle cell actually has its own wrapping, and its wrapping is called number 3, the Endomysium. Now I've placed a little blue covering around the outside of that cell. That covering is to help us understand that there is a sleeve or a wrapping around each individual muscle cell called the Endomysium. Here you can see skeletal muscle cells in this region right here, and you have a perimysium wrapping that covers or encapsulates that grouping of muscle cells or muscle fibers. Let me mention number 4 to you, the term tendon and aponeuroses. You'll notice that tendons are actually kind of an extension of the endomysium or perimysium, as is an aponeurosis. The tendon is actually a connector between a muscle and a bone. So you'll see in this picture the Achilles tendon does grow out of the end of the gastrocnemius muscle, and it connects down here to the calcaneus, which is our heel bone. So we know that these are made up of, say, dense regular connective tissue. So that's connective tissue. The aponeuroses, well, probably one of the best ones for us to talk about is this one here on our skull. It connects the frontalis muscle to the occipitalis muscle. And this region up here, of course, is a layer of connective tissue. So if you've ever seen a bald man wiggle his back of his skull, get that skin to move over the top, it's because the frontalis and occipitalis are connected by that piece of connective tissue. So just a couple of terms here to keep straight. We've talked about the word muscle fiber being the same thing as muscle cell. We do use that term interchangeably. Of course, muscle fibers do include multinucleate cells that are fused together into one long fiber. At the very bottom of your page, under microanatomy, let's talk about number 1. Number 1 is for the term sarcolemma. That's this term here. Sarcolemma is just another fancy word for the plasma membrane of the muscle cell. Sarco as a prefix means flesh, so just keep in mind that when you see sarco, you're talking about muscle tissue. If you read along with me, the next blank there by number 1 is this one, sarcoplasm. Keep in mind there's that sarco prefix. This is just a fancy word that means the same thing as cytoplasm. All we're doing is saying it is inside of a muscle cell by using that sarco prefix. So in this picture, you can see the plasma membrane on the outer surface, and you can see myofibrils, which we'll talk about in just a moment, on the inside. The plasma membrane actually forms these indentations called transverse tubules or T-tubules. So here outside is your plasma membrane, and what's the other name for it? Its alternate name is sarcolemma. Okay, now it forms these indentations. We're going to follow one right in here. It's called a T-tubule, and that T or transverse tubule runs deep into the muscle interior. The purpose of this T-tubule, we think, is to allow nerve impulses to travel immediately to those myofibrils so that muscle contraction can occur simultaneously in these muscles. You'll also notice that this T-tubule is sitting close to another organelle called a Sarcoplasmic Reticulum. We'll talk about that one in just a bit. Now, the last blank on page 77 is for myofibril. So the myofibril is the organelle inside of your muscle. It is a long, skinny organelle, and it has these light and dark striations. So number 2, bottom of page 77, is for the term myofibril. Now, I'd like to go ahead and introduce another organelle here called the SR, or Sarcoplasmic Reticulum for short, SR. This particular organelle wraps around your myofibril like a sleeve. It holds calcium ions, stores those ions, to be released during muscle contraction. So when the muscle is resting, calcium is stored in the SR, but when muscle contraction occurs, the SR releases calcium. That's how we regulate and control muscle contractions, is where we keep that calcium either in the SR or let it leak out into the sarcoplasm. So here you can see the SR. It forms a lacy sleeve around these myofibrils. Let's take a moment to take these words and place them in order from superficial to deep. The outermost covering is Epimysium, this term here. The Epimysium wraps around individual fascicles, which are covered by their own perimysium. So a perimysium wraps around a fascicle, which is a group of muscle cells. The fascicle includes muscle cells, but we know that each one of them is encapsulated by their own endomysium. The endomysium is like having on a long sleeve jacket. So that long sleeve covers over your skin, and you might think of the skin of the muscle cell as the sarcolemma. So let me draw it this way for you. If this is your arm, that's your skin, that's actually your sarcolemma, right? Now if you put a jacket on, you put a sleeve on around the outside, and that sleeve is endomysium. See how that works? Okay, and if I were to look inside my muscle cell, I would find long, skinny organelles in here, and they're called myofibrils. So, of course, sarcolemma is the plasma membrane of your muscle cell, and inside your muscle cell we have myofibrils, which are organelles. All right, so I'll let you look at this one here. Let's now turn over to page 79 and 80. 79 is for us to label together, 80 is already pre-labeled. These are the terms that we're going to cover on pages 79 and 80. Now, let me tell you that on page 81 is where all this information comes from. So instead of having to keep notes and keep up with me, you may want to have all 3 pages handy so that you can look at this information. I encourage you to label page 79 with me as we go. We're going to first look at the types of proteins found in a myofibril. A myofibril contains proteins that form the structures that you see in the bottom picture. We know a myofibril is the organelle inside the muscle cell, and probably one of the easiest things to spot about this picture on the bottom, which is the detail of the myofibril, is this zigzag-shaped Z-line or Z-disc. Okay, and that Z-line or Z-disc is going to be a membrane that anchors a special type of filament. The filament anchored there is called this thin filament, otherwise known as an actin filament. Okay, so you can see over here another Z-line. You can see more thin filaments right here, or actin filaments, anchored to those Z-lines. Now there's a second type of filament. It's this thicker one here, and of course it calls itself the thick filament, and it is made up of a different protein called myosin. Now, because they're thicker, they're easier to spot, but they also have these little knobby heads sticking off the top here. Those little knobby heads are called myosin heads. So we're building our conversation for how muscle contraction occurs. In order to understand how a muscle shortens, we have to understand these micro components. These, of course, cannot be seen with the naked eye. So let's review. We have Z-lines, thin filaments or actin filaments anchored to them. We have thick filaments not anchored to the Z-lines. Those thick filaments are made of a protein called myosin, and they have knobby little heads called myosin heads. Let's now look at this clean picture and talk about the light and dark striation or banding pattern. One of the easiest things to do is locate that Z-line one more time. And this region right around your Z-line is called an I-band. I'm being really careful not to catch those thick filaments in there because an I-band is actually the light colored band that you see when you look at muscle tissue. So if you look at this one up here, you've got a dark colored A-band. You've got this region here that's an I-band. You've got an A-band. You've got an I, and see how it alternates back and forth. So the I band is in fact the light band of the banding pattern, and I remember that because it has an I included. Let's go ahead and circle this I band over here. Okay, label it as an I band. And then let's also include this area here. This area is a different kind of band. It's called an A band. And A-bands are dark bands. They're dark because of the appearance of those thick or myosin filaments. I-bands are light because they do not have the thick or myosin filaments. So keep in mind that dark bands are A-bands. Now, there's a special area inside this A-band that does not have any thin filaments poking in there, and that region is called the H-zone. Okay? So when muscle contraction occurs, those myosin heads will pull along those thin filaments, and those thin filaments will start to invade that H-zone. So we're going to find that during muscle contraction, the H-zone disappears, the I-band will also disappear, because these Z-lines are moving closer to the center of your sarcomere, thanks to the action of those myosin heads reaching up, binding and attaching to those thin filaments. One more thing, let's label sarcomere, which is a distance between the Z-lines. Okay, Z-line to Z-line is known as your sarcomere. So let's review. I-bands include thin filaments, Z-lines, no thick filaments. H-zone is inside of an A-band. An A-band includes thick filaments and some thin, but the H-zone is special because it includes no what? No thin filaments. Okay, so a sarcomere runs from one what to the next what? Runs from Z-line to Z-line. Okay, so that is our unit here of the myofibril. So what you're looking at is a myofibril, which is an organelle inside of a muscle cell. Now, you may want to compare what we've just done to what's found on page 80, and all this is written out for you on page 81. Here again, you can see the Z-lines or Z-discs, which form a zigzag shape here. You can see these thick filaments. You can see the thin filaments right there. Okay. We can also find the H-zone, which is a special area inside your A-band. What's missing from the H-zone? No thin filaments. Now we can look a little more big picture. We can see in this one, you've got the sarcoplasmic reticulum wrapping around the myofibril. What does the SR store? The SR stores calcium. Okay, and of course that's going to be important in our next story for understanding the excitation-contraction coupling mechanism. Here again is another look. You can see nicely that sarcomere is the distance between one Z-line and the next Z-line. That is our unit of measure for the myofibril. We're going to watch the sarcomere shorten as the muscle contraction occurs. So we know that term there is called contractility. Really cool picture, micrograph here of skeletal muscle. Can you find the A-bands and I-bands? Well, what color are the A-bands? These are A-bands. They're kind of a dark purple color. And the I-bands are, of course, lighter pink, and they have a Z-line running right through the middle, which is really cool. So you can see the distance between one Z-line and the next Z-line there is called our sarcomere. Another really cool micrograph of skeletal muscle tissue. Now, can you spot, say, some Z-lines? I'm going to draw one muscle cell here. That is a Z line, and that is a Z line. Okay, so the distance from one Z line to the next is called what? Sarcomere. And the region right around, right between these two lines here, that is called the what band? That is an I band. Okay, here's another I band down here. And in between this area right through here, that's our A band. And you can see a very faint H zone right through there. Okay, that's of course where there are no thin filaments. Now, I know this is a relaxed muscle because I can see visible I bands and an H zone. Had this been a contracted muscle, those would have disappeared because the sarcomere shortens and the Z lines move closer together. In fact, you can see here in this picture, the relaxed muscle at the top has visible I bands, and it has a visible H zone right there in the center of that A band. In a contracted muscle, I have a very hard time finding an I band because it has been basically, invaded by these thick filaments, which butt up against the ends of those Z lines, and I no longer can find an H zone in the middle. In fact, those thin filaments have now invaded the A band, so I have no longer a place where I can say that I've got a space for an I or an H zone. I do want to give you a little more detail here on thin and thick filaments. This is how a thick filament looks up close. You can see the myosin head here, right there, and it kind of acts as a snake. If a snake is going to bite something, it cocks its head and then it strikes at its prey. So that's kind of how the myosin head works. It cocks into this position that will eventually bind it to a thin filament. Thin filaments are more than just actin. I do want to tell you that. Thin filaments actually include 3 types of protein. And if you remember AT&T, that's how you can keep it straight. A, of course, for actin. There is troponin and tropomyosin. So the troponin complex here will bind calcium. That's what's going to allow for us to have muscle contraction take place. This rope-like tropomyosin actually covers up binding sites. You can kind of see them hidden back there on this actin filament. So tropomyosin acts like a blindfold. It covers up binding sites for the myosin heads, and as long as those binding sites are covered, you're not going to have muscle contraction take place because the blindfold is in place. So how do you move the blindfold? You have the troponin complex here come over and bind some calcium. Do you remember where the calcium is stored? It was stored in the SR, right? So when calcium binds troponin, this rope-like tropomyosin is then allowed to move out of the way, like this. See the top picture? The rope-like tropomyosin covers up those binding sites. See here? They're covered and they're hidden by that rope-like tropomyosin. But as soon as calcium is available here, comes from the SR, remember, that wraps around the myofibril, we then can allow calcium to bind troponin, and troponin is like a guard shack. It moves the blindfold off of those binding sites, and now these binding sites are revealed. And now myosin heads can reach up and form a cross bridge with that actin filament. That's how we get the process rolling for the muscle contraction to occur. When you no longer want muscle contraction to occur, you get rid of calcium, and where do you store it? You put it back in that SR, right? And then what happens to the rope-like tropomyosin? It will recover those binding sites, and if you recover those binding sites, can you have muscle contraction taking place? Nope, so muscle contraction then is done. Now, you can pick up with me in our next video. I am going to walk you through how to draw this out. So you may want to watch how to draw this or the sliding filament theory as your next video.